I'm so happy to be here. My name is Katina Calakos. I'm a staff scientist on the Simons Searchlight research team. And I'm excited to tell you a little bit about Simons Searchlight, what we do, who we work with. And that'll be like the first half. And then the second half is going to be what we've learned so far from the data we've collected from all of you generous families spending time contributing data to better understand DLG4. So Simons Searchlight is an international research program with the goal of accelerating science and improving lives for people with rare genetic neurodevelopmental disorders. And we have, I am biased, I'll say that, but I think we have a really awesome team working behind the scenes, ranging from clinical scientists to basic scientists, communication specialists, engineers, really all working to hopefully create a positive research experience for you and to generate important research resources for other people to access and analyze the data that we create, that we collect. And additionally, we have our Simons Searchlight Community Advisory Committee. This is an awesome group of folks who, for 2 years of their time, generously contribute really great insights, a really range of expertise and connections to the different genetic communities that we hope to serve. And so this is who's on our current committee here. And as I mentioned, our mission really is to shed light on these rare genetic neurodevelopmental disorders. And we do that by collecting high-quality, standardized natural history data and building strong partnerships between researchers and families. And so from left to right, first we try to collect detailed medical and behavioral histories along with optional blood samples, which you heard about. We'll be doing some blood draws tomorrow. Additionally, we then synthesize that information that's provided and we share results back with you, the families, both as a token of our gratitude, but also to share back what we're learning as more and more people are submitting data. We also share de-identified data and samples with qualified researchers because we really want as many professional eyes as possible on the data. They can access it, analyze it, publish on it in the literature, and then more people are learning about DLG4. We also don't want to constrict just to our research. We really want to help facilitate other research opportunities if that is of interest to you as DLG4 families. So we have a program where we can connect researchers and participants from around the world. And ultimately, all of this is to promote better understanding of the genetic variants we study, including DLG4. And so we are very fortunate to be fully funded by the Simons Foundation Autism Research Initiative, or SAFARI, whose mission is to advance basic science in autism and neurodevelopmental disorders. And this is also housed within a broader nonprofit organization called the Simons Foundation, also focused on basic science and mathematics. And so the beauty here is not only can we approach things from a genes-first approach, looking at genetic conditions, but we also have partnerships with other research opportunities like SPARK, which focuses on autism, Autism BrainNet, which is a postmortem brain tissue donation program. SAFARI additionally provides a lot of funding for other researchers, ranging from basic scientists to clinical scientists. And then much of the research data that's generated from these collaborations and projects can be made available for additional researchers to access and analyze. So that can look like animal models, cell lines, and different databases where we try to centralize information and make it more accessible. And as I mentioned, we do cover a lot of conditions. We have over 180 conditions now that we're studying, and DLG4 is right there in the red box. I know it's a little hard to see. Sorry about that. The font's a little small. So we can not only really deep dive into DLG4, but then also see, what is unique to DLG4 and maybe what's shared with other conditions, either at the biological level or the clinical level. We also make an effort to expand the opportunity to participate in Searchlight through multiple languages. So I will say most of our measures all of our measures are in English, and then some of our measures are in languages such as German, Spanish, French, Italian, Dutch, and Portuguese. Some of our standard measures, because they're generated by other people and companies, some of those can't be translated, but we are making active efforts to translate the surveys where we can to enhance that inclusivity globally. And so I keep talking about surveys, and it's like, what are these surveys? So here's a whole list of what we administer online. Again, this is all participant and caregiver reported data. At the very top, it all starts with learning a little bit about background history and really importantly, that genetic laboratory report. So we have a team of genetic counselors who thoroughly review that report, make sure you're in the right place. We have the exact details of your variant, follow up with any questions if the report has something missing or they need more information. And that is what unlocks participation in the rest of the study. And so that includes all of these additional measures ranging from medical history, medication use, developmental and psychiatric diagnoses, seizure history. We have a couple of measures on sleep, and then a whole host of measures on development and behavior. Some of these will be familiar to you. We have the parent-caregiver form of the Vineland. We have a new measure out now called the Observer-Reported Communication Ability, or ORCA, looking more at expanding our understanding of communication, particularly for those that are more minimally verbal or nonverbal, behavior, other communication measures, quality of life, et cetera. And because we really want to understand DLG4 at younger and older ages, we ask for families that are assigned these to complete when you first register, and then for a subset, we ask for annual updates to really get that longer-term picture. These are a couple of survey highlights for folks who have been with us for a while. We just wanted to draw your attention to that. We have a new annual medical history survey. The goal here was really to streamline so that baseline and annual updates are a little easier. We preview some of your responses now, so you're not having to tell us the same thing each time because we recognize it does take a lot of time to complete these surveys. And as I mentioned, we also have our new ORCA measure that's available on participants' dashboards. Which, speaking of, if you're in Searchlight, this will look familiar. If you're not, just wanted to draw your attention to when you register and create an account and are eligible, this is what it looks like. This is where all the data magic happens. The middle here is where you'll see the surveys that are assigned. The top being, if you have 5 minutes today, what would we love to hear from you about? And then as a token of our gratitude, we also provide online gift cards at the end of completing those surveys. Again, sharing information back is really important to us. We, again, not just to show appreciation, but really to provide, in hopefully an easy to understand way, some of what we're learning about DLG4. So this looks like some reports on your actual dashboard. This looks like summary snapshots looking across all DLG4 families. So this is a screenshot of the quarterly report that we published online a couple of months ago. This is on the simonssearchlight.org webpage on the DLG4 page. And then in-depth presentations like this at conferences and meetings. We also will do things like virtual webinars, because we know that hosting big meetings like this can be a lot of work. So just let us know if there's any, other opportunities to share. And the Research Match program, I wanted to call back to that opportunity where we want to ensure DLG4 families can participate in other research if it is of interest to them. It's not required by any means. But if there are researchers here or online who are interested in recruiting DLG4 families for other opportunities, this is a great program to do that. You reach out to us. We see how we can try to facilitate that connection, and then the researcher will take it from there. So we've had over 3,000 Searchlight families across our cohort participate in this opportunity. Blood samples, very important for this conference because we will be doing some on-site blood draw collection tomorrow in partnership with Combined Brain. But for those who are not participating in that on-site, or for families who may not be here and are interested in submitting, for those that are based in the US, we do have an option to send a blood sample kit home free of charge. What it looks like is you'd make an appointment at a local Quest Labs to have the blood drawn, and then the lab sends it back to us. But this is currently only available to US participants. And why are we asking families for blood? Essentially, there's a lot that we can learn from blood. We already heard quite a bit from speakers so far. For Simons Searchlight, one opportunity that we currently have that DLG4 has already been involved in is that from blood, we can essentially extract what's called a peripheral mononuclear blood cell, or PBMC. And this is a useful piece of blood because we can take that blood, it's already decided to be blood, we can bring it back to a more kind of infant-like state called a stem cell, and then take that stem cell and ask, can it become something else we want to study, like a brain cell, for example. And this is a really useful resource for researchers to use in their labs because it can help with understanding mechanism, it can help with drug discovery. And so we're really, really grateful to have this program funded by Safari and the Nancy Laurie Marks Family Foundation, where they work with the New York Stem Cell Foundation to identify certain samples that'll be used for iPSC cell generation that can then be accessed by researchers. So I'll talk a little bit about the DLG4 cells. I'll say that it's not a guarantee, so not all blood samples do become stem cells. It's really based on the scientific and clinical priority and lots of conversation, but we are fortunate to have this program. And how's your data used? Fortunately, we've had many publications looking at Simons Searchlight data. This ranges from publications diving into a single gene, looking across more than one gene, the research match studies, and at the very bottom, my soft spot is a lot of the dissertations and theses from trainees in grad school and medical school, kind of helping to try to facilitate some of that more early investigators that are coming into the field and can hopefully stick with DLG4 and other neurogenetic science. And so I'll note for the researchers in the room and online, I wanted to say that we have a brand new data set out as of last month. This includes data from 62 DLG4 families, which is much higher than the last data set we put out, so I really wanted to draw attention to that. Includes baseline and annual updates. And then additionally, there's a new RFA out from Safari called the 2025 Data Analysis RFA. And this aims to provide researchers with the support needed to allocate time and personnel towards working with and publishing from previously collected data across autism and neurodevelopmental disorder cohorts. And priority for this will be given to applications that use Safari-supported resources, including Simons Searchlight and other data sets that are funded by Safari grants. So just wanted to plug some of those research opportunities as well, since I know we have a mixed audience. And one last thing about that, all of that stuff happens through base.safari.org. The Safari RFA is separate, but in order to request resources, researchers can come to this webpage, you create an account, you make a request, and then it is reviewed by the team, usually within a couple of weeks. If ever there's an issue or a hiccup with that, you can always email the team at sdbr@simonsfoundation.org. Okay, so hopefully I've kind of set the understanding of Searchlight and the different resources and to kind of paint a picture of why we're asking for so many survey data. And so now I'll dig into what we've learned so far in DLG4 community. So this is our funnel metrics slide here, which I love to see these numbers increase every time I check in on them. And so for DLG4 specifically, this is looking across families with likely pathogenic and pathogenic variants, as well as variants of uncertain significance, because we do collect data from all three groups. And starting from left to right, we have 76 families that have signed up online. We have 65 who have provided that genetic laboratory report, and a huge thank you again, because that really unlocks the rest of the participation. We have 51 who've contributed medical history data. Another big thank you, because we know that form is long, so we appreciate it. 59 families have filled out additional surveys, and then 16 have provided a blood sample so far, and then we'll have some updated numbers, of course, after tomorrow. So this slide here, so we've already gotten some genetics background earlier this morning, and so they are the experts, and so I won't dig so far into the details here, but I did want to show you just generally what this is showing. So we have 47 DLG4 likely pathogenic and pathogenic variants represented in Simons Searchlight. We are not showing 3 deletions and 15 variants of unknown significance. To the left, top left, is our legend of how we're color coding a little bit. And so what the schematic is showing here is essentially along that protein product that was discussed earlier, what locations are the variants impacting that protein, and what is that type of mutation? So you already heard the different kinds of variant types. Those are color coded at the top. We have some missense variants. We mostly have frame shift and nonsense variants in DLG4, a couple of splice site intronic variants, and then a couple of synonymous variants as well. And so these text boxes here are essentially showing a little bit about the information of how that variant is changing or how that amino acid is changing. So hopefully this gives a sense of kind of the variability in the spectrum of variants that we have in Searchlight so far. As I mentioned, by virtue of being online and with GDPR, we are able to be global. And so we have DLG4 families across the globe. We have 10 countries represented for DLG4, 61 participants. We are predominantly US-based, so you'll see that number is higher, 37. In terms of biospecimens we've collected so far, as I mentioned, we have 15 participants who have contributed biospecimens with a high enough sample that was able to be stored in the biorepository. And from there are 17 samples that can now be distributed to researchers to study DLG4. So this includes 5 iPSC lines and 12 whole blood DNA samples. And speaking of the iPSCs, this is a more detailed overview of which variants were selected for reprogramming into the stem cell lines. So we have 6 variants represented across 7 individuals, since there were 2 folks who had the same variant. And that status column is showing what's currently available. So 4 of those are available now for researchers somewhat recently, actually. So for researchers who are interested, please look into this. We had a couple where we tried to reprogram, but creating stem cells is really quite challenging, even with a rigorous approach that we take. So a couple did not fully reprogram, and we don't currently have more source material remaining. So that is the status of the iPSCs for DLG4. And then the last section of this presentation will talk about some of the clinical data that we've covered in DLG4. We'll primarily be talking about our medical history data and a little bit of development. But as we collect more data, we can do, other spotlights on things like sleep. As I'm hearing more and more about kind of what's important, that would be a great deeper dive to also have when we have more data available. So we have 35 individuals with, all the clinical data will focus on the pathogenic and likely pathogenic variant families. So we have 35 individuals who've given us medical history data so far that range from 23 males to 12 female individuals, early age from 1 year through to 35 years of age, with our average age in this community being 10 years old. Many of the slides will look something like this, so just to orient you, because I live in graphs all the time, but I know not everybody does. What they'll look like is on this vertical line, this y-axis here, or just the vertical line, we will see different kinds of conditions or issues that we ask about in our different surveys. And then the bar will show how many people are reporting having ever experienced that condition or been diagnosed with a condition. And so the longer the bar, the more people are reporting that particular condition. So if we dive right into the middle here, for example, almost everyone who reports to us reports having had some intellectual disability or developmental delay. We had 32 of the 35 individuals reporting some language delay or impairment. We have some folks reporting diagnoses of autism, ADHD, and anxiety, although those are fewer compared to the longer bars above. I know the numbers are probably hard to see here, but got like 46% autism, 43% ADHD, and then a couple of mentions of OCD. If we're looking sort of top to bottom, medical somatic issues, the way this is oriented is the medical or body systems at the top overall are being reported as less affected in DLG4, whereas those at the bottom are reported as more affected. So we're seeing kind of a handful of conditions related to, kidney and heart function, for example, but not too much there. We're seeing a little bit of higher numbers of things related to growth, endocrine, autoimmune, orthopedic, but we're primarily seeing reports related to seizures, vision conditions, GI or gastrointestinal conditions, and then those that fall in the neurological space So for our GI condition, I'll get a deep dive into a couple of these, but for the ones that I don't, like GI, we're seeing GERD and constipation reported most commonly. So we're seeing some hypermobile joints, a few instances of scoliosis, and then I'll talk about some of the vision and neuro next. So for our neurological conditions that we ask about, 80% of folks are reporting low muscle tone or hypotonia, followed by some kind of movement issue or movement disorder in about 43% of DLG4 families in Searchlight. And this call-out box here is specifying a little more the kinds of conditions we're asking about. So we have some ataxias, some tremors, some dystonia, and then a couple of other movement concerns that have been reported by families. Some coordination problems in that lower light blue bar, about 30% of folks, and then different other, 1 to 2 off reports of other neurological conditions as well. In terms of our seizures data, in the medical history data at least, 13 people are reporting seizures in DLG4 and Searchlight. I will say on the very next slide, there's a different set of seizures data coming from a different form where we're getting 18 folks reporting seizures. This is just by virtue of the fact that we're collecting it in a couple of places, and we'll need to consolidate that at some point. But in this medical history data where we're asking about seizure subtypes, these are the different subtypes represented in the DLG4 data: grand mal seizure, simple partial, complex partial. As I mentioned, we have a more detailed seizure history survey that's also shorter than our medical history form. So we have a few more folks reporting here, 18 folks with reporting seizures. And this is where we ask a little bit more about the seizure course. So from what we've learned so far, DLG4 families are reporting that for those who are experiencing seizures are starting anywhere between 7 months to 10 years of age, with average age of about 6.5 years old. At least 90% of individuals are reporting having ever taken medication for their seizures, but that number is likely higher because sometimes, families may not remember some of the medications, or they may not enter the medications data. Most of those folks are continuing to take medications to help with seizures. At least 8 individuals are reporting having achieved a sense of seizure control or remission. The average age of that seizure control reported is closer to 8 to 9 years of age, ranging from 18 months through to 13 years of age. Vision issues, calling back to our medical history form, we're seeing a number of different vision-related concerns being reported: crossed eyes, a mix of nearsightedness and farsightedness, and then a couple of other less represented vision-related concerns. Less affected in DLG4, but wanted to note, since we're seeing it in a couple of families in the data, things like early puberty, a couple of instances of obesity, thyroid concerns, failure to thrive, but overall less affected from the DLG4 data. And similarly, less affected, but wanted to flag a couple of folks who are reporting issues of scoliosis and other orthopedic-related conditions, at least in the data we're seeing in Simons Searchlight. We ask a good deal about medication use, things like what medications are being taken, for what, how effective are those medications, or did you take someone off the medication and why? So there's more medications data in the actual data set than's being shown here, but just to give a high level of medications data, we're seeing at least 44% of individuals are reporting antiepileptics for seizures. Lots of folks reporting medications, about 30% reporting meds used for helping with sleep or GI issues. We did ask caregivers if there was a medication that seemed to work best for their dependent seizures, and no single medication stood out as most effective, but understandably every family is working with their clinical care team to address their own unique seizure situation. And in the last couple of slides, I wanted to show a little bit about our Vineland data. I do want to take a moment to say that we recognize that the parent-caregiver form for the Vineland is long and can be exhausting, and then we ask for it again. And so we are really grateful to all of you for sharing that information, because it really does help paint a picture of development over time and across the DLG4 community. And so what we're showing here in the next couple of slides will kind of look like this, are these growth charts If you saw our quarterly report that we put out last quarter, it looked like this. So that's also available on the Simons Searchlight webpage. But essentially what we're showing here is at the very top is a particular clinical domain or behavioral domain that's covered in the Vineland. In the chart itself, along that horizontal x-axis is the actual age of your DLG4 loved one at the time that you reported on the Vineland. And then on that y-axis or vertical line here, we're trying to get an estimate. It's called an age equivalent. It's essentially an estimate to say, given the skills that have been reported so far in your DLG4 loved one, what is the age of other kids without DLG4 that have that sort of skill set? And it's not a one-to-one per se. These are estimates. It's just to get sort of an estimated picture of growth and development over time. So for example, in expressive language, that orange thick line is showing advancement in expressive language skills over time. Every single data point is a single person at a single time. We have also collected longitudinal data, which is not fully represented here. That's one of our goals, is to figure out a way to kind of better illustrate that within person change over time in future reporting. But for now, we're seeing with our age equivalent estimates, and again, this is not an exact because every family is different, but given the overall skill set we're seeing in DLG4, at least 2/3 of individuals are likely to be using some language at age 4 and older, with many DLG4 individuals developing advanced expressive language skills. We're also seeing in terms of personal care, this is things like doing things for oneself, feeding oneself, dressing oneself. That orange line again is coming upwards. We're seeing closer to 60% of individuals, based on what we know right now, are likely to be feeding themselves at age 4 and older, and there are other people who are already developing skills that are more advanced in personal care. In terms of social development, making eye contact, choosing friends, engaging socially with others around them, we're seeing at least half of individuals with DLG4 are likely to be making eye contact at age 4 and older, and many of our DLG4 individuals are reporting more advanced social development skills as well at younger and older ages, given the variability in the different blue dots representing people. And so overall, to summarize what I've shown so far in our data, we're seeing more commonly reports of intellectual disability and developmental delay, language delay and impairment, vision issues, some neurological issues, particularly low muscle tone, movement, and coordination issues are coming through more commonly. We're seeing GI issues like GERD and constipation coming through more commonly. And then other issues that affect some DLG4 families, but not as many as that first column, are things like autism, ADHD, and anxiety. The seizures data, which I know we've already started talking about, and we'll understanding sort of the differences in seizures between DLG4 families is important. Some orthopedic issues, and then the adaptive behavior and development from the Vineland data. And so overall, I hope that this has been an informative presentation. If you're interested in joining Simons Searchlight, we have a great team. We've been here for a while, and we really want to help invest in the patient organizations like here, and broadening that research enterprise across to different researchers and patient advocacy group leaders. We hope to share data back and continue to share data back with you in presentations like this and other modalities. And we really do want you to represent yourself. The more numbers we have, the better we can understand everyone with DLG4. And so with that, thank you so much for listening to me. Thank you so much, DLG4 Shine, for having us speak and for the blood draws tomorrow. Families can register at Simons Searchlight. Researchers can request data at base.safari.org, and we will have a table with our blood draws at 8 a.m. to 2 p.m. tomorrow. Even if you're not doing a blood draw, you can come over or say hi, and we can talk about Searchlight. So thank you so much.